Welcome to Up One, the podcast where we take a deep dive into the science behind supplements, therapies, and diagnostics. I'm Sawyer Stone, your guide through the maze of health claims, here to ask the big questions.
And I'm Dr. Bill Andrews. With decades of experience in medical research, I've dedicated my career to uncovering the real science behind disease, aging, and human health. On this podcast, we don't just skim the surface of scientific studies. We conduct a critical meta-analysis, separating credible research from misleading conclusions.
There's a lot of noise out there, conflicting studies, bold claims, and endless marketing. Up One is here to cut through it all and bring you science-backed insights that you can actually trust.
We're talking prevention, diagnostics, treatments, and the big questions shaping the future of health.
If you're serious about understanding the science of health and longevity without the fluff, let's take it up one. Dr. Bill, how are you today?
Pretty good, and good morning to you. Actually, it's morning for me, but I think it's afternoon for you.
Yeah, technically.
Yeah. And so I'm doing good, and you?
I'm doing pretty well. We've got some really lovely fall weather over here in North Carolina that's like teasing us a little crisp, like upper 50s, lower 60s in the morning. It's pretty great.
Yeah. Last year and the year before, it was in the hundreds here right now this time of year, but now it's going to be a high of 70 today, which is pretty low for us.
That's so much better, for sure, for sure. Well, Dr. Bill, been listening to our podcasts as they come out just to get a good point of view about what's going on once it's all put together. But I feel like we could play a drinking game, like take a shot every time I hear somebody say critical meta-analysis. And I think that we might be a little drunk if we did that, because we're just saying critical meta-analysis, critical meta-analysis, critical meta-analysis like a lot. And it's not the easiest sentence or phrase to say. It's like pretty bulky, but we should leave that to our listeners.
CMA,
we should not.
Something people
CMA.
Say. CMA, yeah.
CMA. Well, we should leave the drinking game to our listeners so we can be, cognizant of what's going on the pod. But have a couple questions about critical meta-analysis for you if you are up for answering them.
Yeah, go ahead.
All right. Why is critical meta-analysis, or CMA, stronger than a standard meta-analysis, and what should we be looking for when one is done well versus poorly?
Well, typically you don't read results of critical meta-analysis.
Okay.
And so it's not really like what to look for. It's a kind of thing that you would want to do yourself. Okay. But it requires a lot of skills in a lot of areas to be able to do that. And so sometimes it's better to find somebody that's skilled in doing that and get them to do it for you. And I can name about 10 people that I know of that are really super good at critical meta-analysis. But the real problem is that even in the scientific peer-reviewed literature, there's a lot of, excuse the expression, crap that falls through the cracks, and you can't believe what you read, okay? And well, you can't, people believe it. That's the problem, is that people are like redders. Redders, the definition of redder is somebody skilled in the art of persuasion. So people can get publications published because they fool the reviewers, or sometimes the reviewers are not qualified to do in the review, or it's like a lot of times papers are rejected because the reviewers also don't know what they're doing and feel like they'll lose their job as a reviewer unless they find problems with something that doesn't have problems. There's all kinds of different things, but you can find, it's a problem, is you can find anything that you wish to be true in the scientific peer-reviewed literature, because
I see.
A lot of companies will be marketing products and they'll say, well, we need a scientific peer-reviewed study that we can say supports our product. And it turns out you can find a way of getting anything to slip through the cracks. And I've seen this, especially in my own field of telomere biology and telomerase. I've seen this happen a lot of times. There's one case in particular that I actually, so people can go to YouTube and look for videos of me speaking at medical conferences and sometimes general public audiences, things like that. I like to show a really good example of two different scientific peer-reviewed studies in the same journal. I think the year was 2000 is the example I use. The same journal, and the titles are exactly the same. Well, what they did was exactly the same, but the results are exactly opposite. Okay, and
Oh,
so yeah, it's the studies were only 2 weeks apart. Okay, from each other
fascinating.
in the same journal. So, but this is an example I use because it's near and dear to my heart, and because of my expertise in the area of telomeres and telomerase, I was able to go and do my own analysis because I said to myself, they both can't be right. Okay,
Right.
they used the exact same cell lines, they did the exact same assays and stuff like that. But one, when I did the critical meta-analysis, I went and actually looked at the data and things like that, I found that one group of scientists totally misunderstood how the protocols work. Okay, so they were like measuring telomerase activity, measuring telomere length, uh, measuring telomere lengths, and I'm the inventor of co-inventor of almost every assay in that field, and I could tell that they didn't know. One group didn't know anything about telomeres. They just bought kits from a vendor and ran the kits, got the data,
Mm.
thought they understood what it meant, and they got the wrong results. Okay, and one said uh, telomerase does not lengthen telomeres, and the other one said telomerase does lengthen telomeres. That was the pretty much the gist of it. You'll see that I put those in the YouTube videos when you talk about those. But the it definitely telomerase does lengthen telomeres, and that was the one. And I'm
Yeah.
surprised that the other one even got published, but it shows that the reviewers didn't know the subject matter either, and that's how they got it proved. But
Huh.
it's a real big problem. There's, as I said, I think already, you can find anything in scientific peer-reviewed literature that you wish to be true. So if you're trying to market a product such as black grapes will make your hair grow, you
Yeah.
can find it. Okay. And that's because, well, you probably can't find that one because I don't think anybody's tried to market black grapes as a hair growth product. But
No, but
it's,
like you were saying about the Diet Coke thing, and you were saying that you could not find that aspartame was a carcinogenic,
yeah,
and so we were talking about that.
yeah. There's also the phosphorus that's in Diet Cokes that is considered to be harmful to your health. But I so far I haven't not been able to find any peer-reviewed studies actually. Let's say I can find peer-reviewed studies, but I can't find any studies that pass my critical meta-analysis. I mean,
I
they
see.
they will they will have a big publication, and sometimes they'll just reference some other study, and then say this study suggests that Diet Coke or ibuprofen or things like that are bad for you. When
Huh.
I really can't find, in both cases, so those are my favorite examples are ibuprofen and Diet Coke because it's like there's every time somebody has a product that's the best selling product, you're going to find all kinds of critics trying to uh, discredit them or
Yeah.
dismiss their properties just so that people will buy their products instead. And that's a real
Yeah.
problem. And it's like, I don't know how to fix it. Um,
Yeah. Yeah.
another thing, even in the research labs, there's this publish or perish kind of phenomena, which makes
Yeah.
scientists publish even when they don't even believe their own data, even when they can't reproduce their own data later. And this is, again, a problem that you have to be skilled at critical meta-analysis to be able to really figure out what some of the titles of my talk are: separating fantasy from fact, or how to know what's real and not real. And that's what I'm really good at is doing that.
So so if these critical meta-analyses can be taken, or if they're written by the wrong people, they can be sort of false. How do we know, in your humble opinion, which of these critical meta-analysis of peer-reviewed studies, which of these studies are trustworthy?
That's really, really hard. Okay. And it's really that's that's why these people that are good in the art of persuasion, the readers I was mentioning before, they can make it so that the study looks really, really good. But it takes somebody who really understands the field to be able to determine that. So the general audience can be fooled easily. I can this is a mission of mine. Mine is to, really educate people how to do this, but a lot of times it comes down to really telling them to find somebody who's good at this and get them to advise you, because they're like you have to know things like how to design the experiment. Was the experimental design of the study perfectly done? Did they understand statistics and statistical theory? Did they really test enough samples? Did they analyze the data correctly? And this is a big problem, is because a lot of people branch out of their own field when they're publishing papers because they want to do a certain test, and they just buy a kit from some company and do it, and then they totally misinterpret the test, or they over, let's see, they think the results mean a lot more than they really do, but it's so there's also, do they come up with reasonable conclusions? Is there common sense and logic involved in those conclusions? And this is like, there's videos where I actually give demonstrations of me doing critical meta-analysis, and how I'll show a meta-analysis and I'll show the number of papers that show one thing and a number of papers show one thing. And then I go through and do my critical meta-analysis, and I can show how the numbers in one column start decreasing, whereas the other one doesn't decrease. And then you're left with all the papers, studies that pass the critical meta-analysis say the same thing. People, you do a YouTube search for a talk I gave in Tokyo in 2017 to an audience of about 500 doctors. That's probably the best example of where people can find that. But again, it's like you have to be obsessed, like I am. On trying to,
it's a special skill.
Trying to be really good. But people can learn, that people can go. In fact, all through my schools, I never pursued a PhD. They just handed me one, because all I was trying to do was learn everything I can to be able to learn about what is really healthy and what isn't healthy, and what extends lifespan and what doesn't extend lifespan. So long answer, typical of what I usually do, but
that's okay.
I think I answered the question. Answer.
Yeah, definitely. Well, okay. So then if you were going to design one of these studies and you were going to make it the perfect study, in your opinion, on some new anti-aging therapy, your favorite, what choices would you have to make about the design, like randomized trials versus observed trials, or what would make or break the credibility of the results that you sought?
Well, first it would depend on a lot of different things. Like, is it a nutraceutical or a pharmaceutical or a diagnostic that's being tested? Is it?
You pick.
Yeah. Okay. So the easiest one probably is a pharmaceutical. Nutraceutical. Okay. So
wait, while you're talking about it, can you tell us what the difference is between a nutraceutical and a pharmaceutical?
A nutraceutical is anything derived directly from plants without making any changes to it. Okay. As soon as if you isolate a ingredient, a chemical inside of a plant, and you modify it to make it better, it's no longer a nutraceutical. It becomes a pharmaceutical. So to maintain its status as a nutraceutical, it has to be something that's purified, or an extract made straight from plants. And so the FDA, when the FDA was first created, there was some rulings that forbid the FDA from putting any regulations on nutraceuticals, plant extracts. because they got a classification as generally regarded as safe. Okay,
Mm,
and uh, that's because people have been taking some of these plant extracts for 10,000 years, and people haven't really noticed any dangerous side effects. Let me just say that the problem with that is that there are a lot of plant extracts that do have a lot of side effects. And
right.
typically over evolution, not evolution, but over time, over the last several thousand years, we've just learned not to eat those foods, those plants that cause, if somebody died suddenly from eating a plant, everybody else say, well, I'm not going to eat that plant.
Yeah.
But it, it doesn't cover the category of plant extracts that kill you slowly or cause damage slowly. So if
Right.
it's a buildup that's going to kill you like 20 years later, um,
Yeah.
nobody's going to know that. And plus, no, humans aren't going to evolve a way to tolerate something like that because once you've raised your young, there is no evolutionary advantage anymore for you living longer. So
Right.
nutraceuticals could be a problem. But the reason why I think it'd be easier to talk about pharmaceuticals than nutraceuticals is because you don't really need to do clinical studies of nutraceuticals. And plus, every time I hear of somebody doing a study on a nutraceutical, it takes so long and costs so much money that when they get done, they find out that the scientists in their company have already come up with the next generation. And
Oh,
now nobody's taken the product that they just tested. So they take the new one.
right.
Okay, because it's so fast to get nutraceuticals on the market. So is it a transitional product nutraceutical or is it a final nutraceutical? If it's a final nutraceutical, which is actually really rare, um,
Yeah.
it then becomes something that's worthwhile doing a big clinical study. But I've always, every time I've heard of anybody doing a clinical study on a plant extract, it's it's like worthless by the time the study gets done because somebody's found some other plant that's even better. Um,
Right.
but so pharmaceuticals. Okay, so in the longevity field, then there's a question of safety versus efficacy. Okay, is the product
Right.
safe versus is the product does the product work? Okay, and then there's also with human pharmaceuticals. Is it an animal study or is it a human study? Okay, because it's sometimes very impossible to do human studies
Right.
when sometimes for a person skilled in statistical theory will be able to calculate, well, I'm going to need 10,000 people for this study and
Right.
they all have to be identical twins, randomized. The twins all have grown up the same way their entire lives. So it's really hard to do sometimes clinical studies on pharmaceuticals, but the animal studies, um,
Yeah.
okay, so then there's another question. Is it my study or is it somebody else's study? Because
It's
personally,
your
I
study
don't,
because you're designing it.
yeah, okay, good. I don't believe in mouse studies. I think that mice, in fact, I've invented a lot of cancer drugs. Um, you know, you hear about me being aging, but I actually have a strong background in cancer too. I was actually National Inventor of the Year for my cancer research. But I've developed a lot of cancer drugs that just work beautifully in mice, but
Right.
fail tremendously in humans. And it's something that there's, again, YouTube videos where I talk about this, especially on a subject called best choice medicine, where I talk about how uh, animal studies are not necessarily translatable to human studies. And I forget the numbers, but I say something like 5% of anything that works in animals actually works in humans. And
Wow. That's really
it's
low.
a very low percentage, especially when it comes to things like cancer drugs. Um,
Right.
the and there's a lot of reasons for that, and I definitely don't have time to go into those right now. But um, I I would I would do my animal studies on pygmy marmosets. Okay,
Oh,
and that's because pygmy marmosets are pretty much the size of mice, so you can do a lot of them. And I'm also very opposed of doing any so animal studies are called preclinical studies. I would be opposed to doing any preclinical studies that brought harm to the animals. Okay,
Yeah.
I'm a very animal rights activist,
Yeah.
but
Which is
I
probably rare as also being a scientist.
actually not true. I mean, every
Oh,
company I've worked
great.
for that did have animal studies, 90% of the scientists were appalled. It was the clinical affairs group that was doing all these things, and they would send them out to other companies like Charles River or something like that to do the studies,
Yeah.
to dissect the animals. And just
Yeah.
I've worked in hospitals where I would walk up and down hallways on the floors where they're doing animal studies, and I was just appalled. It made me nauseous. But so I believe that if we're going to do a study on longevity in animals,
Yeah.
the animals have to benefit from those things. And
Right.
a lot of the studies that you do with animals require dissecting and things like that, supposedly, but you don't really have to. It's like the question is, if you're doing longevity, did the animals live longer? Did they live healthier? Period. That's all you got to really do measure. Like
Right.
when I talk about my preclinical studies with pygmy marmosets, I haven't done yet, but I've already identified places in South Colombia that would do it and things like that. I would have the pygmy marmosets all still in the wild in a netted area, okay, and just have tags
Sure.
on them and just have food for them and everything like that, protect them from predators, and just keep track of when one quits moving, okay, pretty much
Yeah.
with its tag.
Yeah.
And that would be
Yeah.
the way to do the study. And it wouldn't be because of the treatment. Well, hopefully it wouldn't be because of the treatment. But if suddenly the treated mice or treated pygmy marmosets started uh, uh, not moving fast, I would instantly quit using that treatment. Quantitative data is completely worthless without qualitative data. Okay, and so
Okay.
and that's why a lot of the studies. I'm a big believer in qualitative. When you hear me talk at conferences, you'll hear me talk about the Betty White test. Okay, I mean, okay, even in companies that I work with, that and Betty White test. You can look at a picture when she's 25 and 85, and you know which picture was taken first. Why?
Yeah.
Don't know. Is it quantitative? No. Is it qualitative? Very much so. We use a term here at Sierra Sciences called QQ scores, which means qualitatively quantitative. Okay. So
Mm.
what we do is if we're trying to find out if a skincare product makes your skin look better,
Right.
we do measure wrinkles and we do measure blister formation and things like that. But more importantly, and I think the most important test is to have people look at a computer screen and see two pictures and then
Right.
say, which picture looks younger? Okay. And then click that one. Then two other pictures. Which picture looks younger? Click that one. Which pictures look younger? Click that one. That kind of thing. And that's all qualitative. But I think that's so much more important than anything else, because when you buy a skin product, you don't want to know that your telomeres are getting longer or your DNA methylation patterns are changing or uh, your IgGs are getting glycosylated in different ways, different markers of aging and longevity that people use. You
Sure.
want to know that your skin look better, and you want to
Yeah.
know that when you walk down the street, everybody looks
People
at
are
you and thinks
thinking,
so.
gosh, she
Yeah,
looks great.
yeah, that's exactly, and that's the most important thing of all. And so
Yeah.
I've written clinical protocols, a lot of them. I have some that are like 200 pages long for looking at various techniques, mostly pharmaceuticals, because I wouldn't spend that much money and time on a nutraceutical, especially when we're going to have a better one coming sooner before the study will be done. But but a lot of it is qualitative, okay? And
Sure.
in the clinical protocols, I identify exactly how to take a before and after photo, okay? And
Yeah.
no makeup, similar lighting, do it in a dark room where the only lighting source is a light that can be consistent from time after time. No
Yeah.
smiling. If you insist on smiling, make certain it's the same smile each time. lots of different things, because the qualitative is far more important. But still, so in designing the perfect experiment, and the clinical study, let's say we've already been through animal studies. Now we're doing humans.
Yeah.
I would be doing 100 different biomarkers, okay? Because I don't believe any biomarker alone is telling you that you have reversed aging or affected aging or even slowed. Well, first of all, it's impossible to measure the slowing down of aging, except in animal studies. So you could do it if you had 10,000 pygmy marmosets in one group and 10,000 pygmy marmosets in another group, you can measure the slowing down of aging. But you just can't do it in humans. I mean, it's like...
well, then how do we know that Telovital is working?
It's faith, okay?
It's faith.
That's what's really, you have to go on faith regarding the biology of the product, okay? We do know that
Sure.
lengthening telomeres does reverse aging in animal studies, okay? We know that it reverses aging in human cells grown in a petri dish. We know that it reverses aging in human skin grown on the back of a mouse. And in order for us to, and then we can even measure slowing of aging sometimes in those cases if we do enough samples. But
goodness.
But to do the studies on humans, that would take an incredible amount of work, stuff like that. And so,
in time,
I always say we resort to what are called phase 4 clinical studies. Okay, so you hear all the time about phase 1, phase 2, phase 3 of clinical studies, but you rarely hear about phase 4. Phase 4 is, and this is practical, it can be done with nutraceuticals or pharmaceuticals, but phase 4 is after the product has already been marketed. Okay, and in the case of pharmaceuticals, it's by prescription only. Okay, so the doctors prescribe it, and then to their patients, and then when their patients come to them with adverse side effects or serious adverse side effects, or adverse events is what it's called, they'll record it and let the manufacturer know. And so that's why when you buy a pharmaceutical, you see these lists of all these different things that can happen. That's comes from those doctors getting feedback from the patients.
Right.
That's what a clinical phase 4 clinical study is done. You can do that on with nutraceuticals too, but now it becomes more anecdotal because nobody's actually doing the actual measurements. That to make it even more complicated, there isn't a chemical or ingredient from a plant extract on the planet that somebody isn't sensitive to. Our immune systems are so different from each other. I often talk about the V, D, and J regions of our chromosomes, which are the regions that are shuffled to create our antibodies. Okay, and everybody's are different. So there's always going to be somebody that's going to be sensitive to something, and so it becomes a numbers game. If like 0.1% of the people taking the product have this serious adverse effect, you just tell those people to quit taking it. Okay.
Yeah.
Nothing else you can do. So in terms of testing in pharmaceutical, I would be looking at blood biomarkers, like especially homocysteine and insulin and a host of others. I think there's like 50 of them in my clinical protocols that I would be looking at, because just reversing one means nothing. Just affecting one means nothing. It just means you're affecting that one particular marker. You got to see all the changes that are associated with aging. So a lot of blood markers, and it includes telomere lengths. And in my clinical protocols, I use more than one method of measuring telomere lengths, but because telomeres are so difficult to measure. But DNA methylation changes, that's easier to measure, so I include that. And also IgG glycosylation is something. Those are the 3 biggest ones. Well, there's also total RNA sequencing, which is super expensive, but it's the best way to test for epigenetics. People call DNA methylation epigenetics a lot, but it's actually not. It's actually a epigenetic mechanism. It's a way of controlling epigenetics. But total RNA sequencing is the best way to measure epigenetics. And epigenetics is the study of genes being turned on and off, because that's the biggest thing about aging is you have
Right.
25,000 different genes in your body, in every cell of your body, and except your red blood cells. You have all these genes, and your parents, your health, your ability to do things is all affected by the relative proportions of how much each of these genes are turned on or off. Every gene has like a dimmer switch next to it that turns it on and turns it off. So that's epigenetics. It's the study of how much is a gene turned on, how much is a gene turned off. And so,
Is that like how some people get blue eyes and how some people get brown eyes?
well,
It's either turned on or turned off.
sometimes that, yeah, in those cases, those are permanent. So there's not a dimmer switch. It's sometimes
sure.
an on-off switch.
Yeah.
And sometimes it's permanently on. But the genes that affect aging are all dimmer switches. And so what you want to do, and there's a lot of genes that are correlated with aging. Even back in the 1990s, when we were studying aging, like skin growing on the back of a mouse, we would find from essentially doing equivalent of total RNA sequencing, which wasn't available then. We would find 50 different genes at least that were changed with aging. And there's a few publications that I'm an author on that came out in the 1990s and things like that about things. But there's a lot of genes that have changed. But those genes, those turning off, that's what gives us our wrinkles and our hair color and things like that are associated with aging. And so we want to measure all those things. And the best way to measure those is from total RNA sequencing. Now, so we also want to be testing physical things like balance. Balance is an extremely important thing. There's a Dr. Joe Raphael in New York that just did a big publication about that, which I was very impressed with. About balance may be one of the best markers of aging there is. But there's also let's see, vision, cognitive skills, hand grip. Hand grip is one of the most important ones. And it's hard to do hand grip studies with animals, but there's ways of doing it by making them climb up through a hole to get their food and things like that. How good are they at doing that? But so I would be doing heart exams, cardiovascular exams, brain exams, carotid artery exams, all the different organs, looking for what are called liver transaminases. Liver is one of the first things that fails when you get older, and when livers start failing, you get release of transaminases from the liver, and you can measure those, and those are things that are included in all the studies. But it's like these clinical studies are super expensive, and we only do them when we actually have a final product. We're not going to do them, and then if we're still developing a new product. And a lot of times it's impossible to raise the money. Sometimes it costs $1 million to test one person after one treatment. And so it's really, but a lot of the big problem, especially with pharmaceuticals, is a lot of pharmaceutical companies cut corners. And I've been
trying to do cheapest and fastest.
I've there's at least 6 or 7 different drugs that I've invented. Well, a lot more than that, but 6 or 7 that actually got approved through clinical studies. But there's others that I invented that didn't get approved, especially cancer ones. But I was never actually part of the clinical study. I just invented the product, but I would always be attending the clinical affairs meetings, getting updates on the testing. And I was always appalled at how many things got dropped, either because it was too expensive or because somebody forgot to prepare for it. Okay. All of a sudden, you're doing your clinical study. I call it the oops factor. Okay. So clinical study be done. Patients, you got 50 patients sitting on hospital beds, IVs in them and stuff like that. Okay, it's time to measure homocysteine, levels. And all of a sudden the nurse comes up and says, "Oh, we forgot to order the kit." Okay, that happens a lot. And so they say, "Okay, let's move forward anyway." Okay, so in my clinical protocols, I'm the orchestra leader. Okay,
Sure.
I'm not an MD. I'm a PhD, but I teach continuing medical education all the time. So and it's no offense to MDs, but they're so busy treating patients.
Right.
Sometimes I have to refer to them as cattle herders. They're one patient out, next patient in, next patient. They don't have time to learn all this stuff. And then when they do learn it, they learn it from sales reps that are obviously biased. So there's such things as conferences, which are continuing medical education, where the doctors go, they actually listen to the lecturers, and I teach at these things all the time. They listen to them, then they're given a test that I'll have written my questions of the test, and they have to pass the test to get a certificate showing that they have participated in it. And they get to hang this in the wall, and now all of a sudden they have a new field they can do.
What randomized control trials are often called the gold standard of trials. When it comes to longevity research specifically, are there blind spots or limitations to a randomized controls trial?
Uh, well, they're really hard to do, but I do believe that the best kind of measurements come from double-blind, placebo-controlled, randomized trials. The critical thing is it's hard to randomize humans because every human is their lifestyles are different and things like that. But
It's already randomized.
and so in order to be able to say that you have two groups that are pretty much identical in terms of certain traits, you have to have a lot of groups. You have to have a lot of people in each group. But in animals, yeah, it's a lot easier to do. But so I would never say it randomized, double-blind, placebo-controlled randomized study is a waste of time. But it does come right down to being qualitative instead of quantitative. But
Quantitative.
it's like, I mean, I try to include as much qualitative stuff as possible, but I try to do, like, if somebody, like, there's been times when I've actually done it publicly, like randomized placebo-controlled studies where somebody is at an actual conference. And like, I remember one in particular at an A4M conference. There was a booth where somebody was had a product that they were trying to promote based on energy levels. And I do believe there are products that work with energy levels, but I also believe there's a lot of quacks and charlatans that take advantage of certain tricks to try to fool you into thinking that their product does different things. And so I remember doing there was this group that had this bracelet that you'd put on your wrist, and then they would show that it's easier to tip you over, or if you don't have the bracelet on your wrist, you can fall over easier than if you had it on your wrist. So I did this, I challenged them. I did a double-blind, placebo-controlled experiment. I first said to them, do you have to have the wrist on your wrist? And they said, no. I said, okay, well, let's put it in a bag, in a brown paper bag, and then just have a person hold on to it. Okay. And so we got some things that felt exactly the same as the wrist. And I had a circle of people standing around wanting to watch the study. And I would have somebody else randomize the bags.
Yeah.
And then we would provide the person, several different people with bags. And then the people selling the product would then try to tip them over. And it turned out, and then afterwards, we opened the bags to find out who had what. And it was completely useless. Okay. And so as soon as we showed this, the person who was running that booth at the conference just started making a scene, like calling me a fake and fraud and things like that. And I just had to walk away from it. But I think I made my point. I think nobody was questioning it. But that was the advantage of double-blind, placebo-controlled, because nobody could accuse me of being biased, okay, in the study.
Right, because you were also blind to which one had the wristband.
But it's rare and so in terms of quality, being able to push over, that's a qualitative thing more than a quantitative anyway. So it's still it's hard to separate qualitative. Qualitative is the most important thing.
Yeah. Okay. So when the researchers are pulling all the evidence together for their studies, a critical meta-analysis isn't just like pulling the results. How does it actually weigh the strengths and weaknesses of different studies to get a more trustworthy conclusion? We sort of talked about this earlier, but like, what is the critical meta-analysis like studying in the study to get the best result and to spit out like what might be the best conclusion or a trustworthy conclusion?
You don't actually do a critical meta-analysis during the study. Critical meta-analysis is done after the study is published or even not published, okay, when somebody else reviews it. Okay, but
Right.
if you are somebody that wants to do a really good study, you have to be skilled at doing critical meta-analysis so that you know that
I see.
it will pass the test. And that's why like my clinical protocols are so expensive and time consuming, is because I design all these things into them. But yeah,
Right.
so you gotta know your animal. It's typically so. I remember in graduate school, there was always this big emphasis that when you're going to do any study on animals, you have to know all about that animal. You can't just
Right.
it's you have to know everything, and because in order to know what to look for. But it goes beyond that. It has to also be, you have to know the particular everything about the particular test you're doing. Let's say telomere. Okay, you're measuring telomere lengths. Um, so many people do it wrong. So many people misunderstand the limitations of various protocols. And that's why I recommend DNA methylation and IgG glycosylation instead, is because it's so hard to interpret. And it's also so easy to make the results be exactly what you want them to be. Okay, because
Right.
it is, I mean,
Because you're running the study.
I want to say in the 1990s, I actually gave a presentation where I showed how I could change, like there's two different methods I was picking on. One was what's called PCR measurements of telomere lengths, and the other one was Q-fish measurements of telomere length. And I actually showed how I could make the results be anything I wished them to be just by slight changes in the salt concentrations or slight changes
Oh,
in the
yeah.
temperatures. And so all the more important to be double-blind placebo controlled, because
Right.
everybody is affected by biases. Okay. And
Right.
the subconsciously or consciously, it's impossible to rule it out.
Right.
Even I've been through so many clinical studies, I still find myself prone to possible things. But it's like
Biases. Yeah.
it's like a lot of times scientists will say, "Well, shoot, this result doesn't make sense. I better repeat it." Well,
Right.
that's exactly the big mistakes that get made. You keep repeating it until it makes sense. Okay, and then all of a sudden the result is biased, and not always because the person doing the study is trying to market a product. Sometimes they're trying to market themselves. And I can tell you, there are scientists I know that have fake data just to increase their own um, popularity in the field. Okay, and this is something that's called
That's not good.
histrionic personality disorder. Okay, so I also have a degree in psychology, mostly because that was the best place to learn about statistical theory and statistics in general. But histrionic personality disorder is a disorder of people that have to have attention. They have to say, "Look what I've done! Look me!" And
I thought that was narcissism.
yeah, same thing. It's narcissism is usually about look how great I look or something like that. But it's it's like there's so many studies out there that from scientists that just are like they feel like they are useless unless they can get something else out there that published that gets in the press and gets a lot of attention.
Well, that must make them biased about their results and what study they choose to do if they're looking only for recognition.
Which actually brings to another problem, is that even when doing meta-analysis, you don't get a paper published by trying to publish something that everybody else has already done, okay, and shown. You only get
Yeah.
it published if you show a study where it's different from everybody else's. And that
Yeah.
tends to now get most of the new publications are the ones that say that it's different. Well, what about the ones that were all identical? You suddenly start ignoring these things, and next thing you know, press releases, somebody has just proved that something we believed all along is not true. Okay, but they're
Right.
wrong. They're wrong. That's where
Yeah.
my critical meta-analysis comes along, and the CMA, and I
Yeah.
can convince myself at least, I would try to do this at medical conferences and things like that, but I've gotten a lot of cease and desist orders. Okay,
Oh.
so it makes it really hard for me to do that. So I tend to kind of
Yeah.
like leave it, you know, to friends and family and things like that. Uh, but or sometimes not. Well, sometimes it's impossible. But I try not to name names in terms of products and things like that.
Yeah. Well, okay. So you just said this, like when we get a big headline, and just this week, I don't know when this is going to come out, but just this week, the New York Times reported that all of us that have been paying attention to our astrological sign for many a millennia are probably thinking that our astrological sun sign is wrong because there was a 13th astrological sign once upon a time. And also there was like the sun rotation changes. And so the day that which the constellation was behind your blah, blah, blah on your birthday and so all this stuff. So, and it disrupts this whole universe that's been created around this idea of astrology. And it becomes like all of these books that have been written, are they now wrong? Is the New York Times just doing an op-ed? Or is astrology so unmeasurable that it becomes opinion? So it's like we get these breakthrough studies and these headlines, and we can't replicate it. Like you can't really replicate where the sun was on your birthday 20 years ago. In aging research, how common is that? And what can a meta-analysis reveal about why replication sometimes falls apart?
Well, okay, so you said headlines about new discoveries and things like that, and that's where critical meta-analysis comes in really handy. But now I have not seen this headline that you just talked about this changes in astrology, but I can say that I don't know if astrology works or not, but
Right.
I'm a big believer in the possibility that it does. Okay, and I
Yeah.
respect people's beliefs in astrology, but it depends on it would depend on what actually what factors did change. Did this did the people who just published whatever something you were just talking about, did they actually show that the sun really was in a position different? And has there been peer review to
Right.
prove that they were right and things like that before it changed anything? But there's all kinds of things that happen like that. And so it's got to be, is it reproducible? But the person that published
Right.
that might just be somebody who suffers from histrionic personality disorder or who's dead narcissist. Okay. And they just needed to get some publication. And the other, it's not necessarily histrionic personality disorder, which I sometimes call HIP, but
Yeah.
or narcissism. It's also because of the publish or perish. I'm
Right.
crossing my fingers. Let's see. Publish. Sorry, I should. It's the publish or perish phenomena that exist in the scientific community, especially
Yeah.
the academic community, where in order for people to get grants and other funding sources, they have to demonstrate a certain number of publications. And so sometimes scientists will resort to anything to get a publication. And again, it's hard to get a publication when you're just showing that everybody else is right. You got to show that everybody else was wrong to get the publication. And
Right.
so happens a lot. So it'd be interesting if I was to do the critical meta-analysis of this astrological stuff. I'd be very curious as to what I end up finding out.
Yeah.
I mean, I've done things like that on other related things like did Methuselah really live to be 900 plus years old and things like that. Um,
Yeah.
and it's really impossible to tell
Right.
because
Because we're not there.
there's a lot of evidence that they were counting years by full moons, which
Right.
changes everything. And can we prove it? No.
Right.
But yeah, but Okay, so things that have made headlines that really turned out to be false. Okay, is all this stuff like all this stuff about coconut oil being a cure for Alzheimer's. Remember
Oh my
that
goodness.
a
I
few
have
years
not
ago?
heard that.
Oh, shoot. Check back on this. There was a woman, and don't even think she was a doctor, but she had cured her husband of Alzheimer's with coconut oil. Okay. And she got all
Wow.
over the news. She wrote a best-selling book on using coconut oil for treating Alzheimer's. Trader Joe's, the grocery store Trader Joe's, started having mountains of jars of coconut oil. When you walked into the store, you almost had
Yeah.
they were in your way. You had to walk around it. People were buying them like mad
Yeah.
because people believed this rhetor, this person
Yeah.
who was skilled at the art of persuasion. She never mentioned that her husband died from Alzheimer's, uh, but and before the book became a bestseller. But if people started figuring it out that this was all they've all been had, and now Trader Joe's does not market coconut oil. And
No,
then plus
same.
there's
Nope.
been no big press release saying it's all fraud. They just
Right,
made
of
it
course.
disappear. Okay. And this woman made a ton of money at everybody's expense.
Yeah.
And that's one of my favorite examples. Another
It'll
example,
be it'll be a Netflix documentary in like 5 years.
Elizabeth Holmes type
Yeah, exactly.
Netflix thing, which I thought was a really great series. Another good example of stuff that really got a lot of press was all these pharmaceutical companies coming out with these monoclonal antibodies that would attack beta amyloid. I know I'm using terms here that there was if you were to look at like monoclonal antibodies for treating Alzheimer's, again, it's another Alzheimer's thing. Everybody thought that because of the fact that when somebody has Alzheimer's, they have this appearance of things in their brain called beta amyloid plaques. And so
Okay.
the question was it a result of Alzheimer's or was it a cause of Alzheimer's? And people like me, I was doing research on Alzheimer's back in the 1990s. And I pretty much was 99% sure that beta amyloid plaques, and all the scientists I worked with, we were sure that these were results of Alzheimer's, not a cause of Alzheimer's. But still, a lot of people kept thinking this is what's causing Alzheimer's. So all these pharmaceutical companies, big race to come up with monoclonal antibodies, which is a kind of like immune treatment that will attack the beta amyloid and cause it to disappear from the brain. And a lot of us were thinking, what a waste of time. These companies, don't they know better? But there was
Yeah.
millions and billions of dollars spent on trying to do this. They all these like 5 different companies came out with these monoclonal antibodies all at the same time. None of them worked. Okay.
Ugh.
And it was like a lot of the scientists were just fuming mad because all this waste of time and expense and marketing and things like that, when it was pretty obvious that it was never going to work. But those kind of things happen all the time. There was so a new possible treatment for Alzheimer's is lithium. I just heard about
Oh yeah, I've heard about this as well.
lithium, but combined with a particular type of salt that allows it to get through the blood-brain barrier. Like I forget the name of the salt.
I don't know about lithium in your blood-brain barrier. That feels like we're going back in time to like treatments at mental facilities back in the day.
Yeah, well, lithium has gotten in the news a lot lately because it's also being hyped as a telomerase inducer to lengthen telomeres. But it could be all false, and this is why I do critical meta-analysis, because I'd like to follow up on these things. But I'm very anxious. Probably as soon as I get done with this podcast, I'm probably going to go and do some critical meta-analysis of this new lithium version, which somebody has published, because the other ones don't get through the blood-brain barrier, so it doesn't really get to the brain. This new one is like 1,000. You can use 1,000-fold less dose and get it into the blood-brain barrier, and it's affecting Alzheimer's. But I'd like to see the data because I've only seen so far the press releases. I think it was late last night that I saw them.
Yeah. Well, this sort of brings me to my next question. You've like kind of answered it, but I'm going to ask the question anyway, and then you can go back and fill in the blanks. Can you give us a real world example of where a supplement or a therapy looked exciting at first, like this lithium thing, but once the data was aggregated, the story completely changed? I mean, I think you were saying that about the coconut oil.
Yeah.
And you were saying that maybe about this lithium, depending upon what it is. I mean, it just seems crazy to me that we like get all excited and we get it all out there and like everybody's hopes get up. And then like all the scientific research isn't done properly and like the expectation falls.
Well, the problem really is that this is very common in the nutraceutical field because nutraceuticals, scientists can take advantage of the generally regarded as safe rule, and they can get publications. But I'm going blank on. I mean, there are like lots of different things that have been in the press a lot lately. And the best way to find it is, was it in the press and it's no longer in the press?
Right.
And
Right. It
that's
just
the best
pops
way.
up like they were talking about how you had to like you could get rid of all of your like mouth cancer if you swished with I think it was coconut oil for a couple times a day. You like mouth pulled, you like never brushed your teeth again and you just did that. I mean, it's crazy the stuff that comes out that's like this is the new best thing that's like going to change your life. Like cottage cheese is on like the biggest high of its whole life right now because people are so obsessed with protein. But in a few months, cottage cheese will just go back to being what it is. It's just these like fads that go in and out.
There are so many things that come into that category. I can't remember them off the top of my head, but if you can think of it, I can tell you eggs. I mean, look at eggs. All the press about eggs are bad for you because it gives you cholesterol, but none of that was true. The cholesterol
Right.
in eggs get totally destroyed in your intestines and stomach and things like that. And it doesn't contribute to the cholesterol load that you have in your body. The sugar is actually what does that. I mean, I think sugar is something that's a permanent thing. We're never ever going to say lots of sugar is good for you. Okay.
No.
But this
Yeah.
whole thing with look at the bad rap that eggs got for so long, and now
They really
they're the
did.
greatest things there are. And part of it is because of the protein. But
Yeah.
protein is it's like one of the things that about protein is that well there's okay, two different categories. Okay, protein, if you're just trying to get protein to get the amino acids to help build all the other proteins in your body, the I you can tell when I get excited about a particular subject. I start tapping my fingers, and I'm sure that's messing up the recording. The but the it really all proteins are really the same. All you got to do
Mhm.
is look at the amino acid composition because because you want to have a well balanced amino acid distribution, and it turns out from an evolutionary perspective, the best amino acid composition to allow us to live long enough to raise our young is pretty much the amino acid composition of meats. Okay. But you can get the same thing from plants. Okay.
Yeah.
And that's why a lot of different plant proteins are a mixture of different proteins because they're trying to make the amino acid composition to be equivalent to uh, whey or meat or things like that.
All right. I just have a couple more questions for you
Okay.
because I know you're booked and blessed. Um, can you walk us through a specific example where researchers mistook correlation for causation in aging science and what the consequences were for the field or even for the public?
Uh, a lot of them. I mean, all the, like the DNA methylation, IgG glycosylation, even telomere lengths, they're all correlations. Okay.
Right.
None of them have been shown to be causative. I mean, telomeres, I think, come out to be the best to be show that telomere length does cause diseases and aging when they get short. Um,
Right.
and but the jury is not really out until we can start finding ways to lengthen telomeres in humans and show that the results, because scientific method involves like the test that you do. You have to first be able to reverse some effect that you see and show that so if you think something is a cause, you got to reverse it to show that the
Right.
cause goes away. And then you have to do something called extinction, where you then have to allow it go back to the way it was to show that it did cause the disease again. Then you got to reverse it again. That's part of the scientific
Right.
method protocol.
Yeah.
But none of that's been done with any of the no biomarkers of aging, because nobody's ever passed what I call the Betty White test, which is really going to be the cure for aging. Now, longevity can also be
So
defined.
wait, when you say that, sorry to interrupt you. When you say that, are you saying that when you say the Betty White test and you're saying, you said this earlier in the podcast, that you can look at the picture and know one picture was taken before the other? Are you saying that in order to pass the Betty White test, you want the same two pictures and the person to say that the picture that was taken after the first picture was taken before the first picture? So you want the results to be reversed, and that this person does in fact look younger in this photo that was taken later in their life, and so that makes them pass the Betty White test. Is that what you're saying?
Yes, but it wouldn't be just looks. It would be look, feel, and behave. Okay. So there'd
Okay.
be several different studies that would be qualitative. But but that's what I mean by that. Because unless you do like 500 different biomarkers and show they all reversed according to things that correlate with aging, you really can't say that did you reverse aging or did you just reverse the biomarker.
Got
So
it.
longevity can also be defined as just living longer. Okay, it's like
Yeah.
an anti-cancer drug is a drug that increases longevity because people live
Right.
longer by taking. But that's a whole different thing. But focusing on just longevity result of aging, there's nothing that's not a correlation. They're just a correlation. Everything there's no because until somebody actually demonstrate reversal of aging by what I call the Betty White test, we don't have any measurement except correlation.
Yeah. Okay. Well, when you are planning a study, how do you determine the sweet spot for a sample size, like enough people to see meaningful effects, but not so many that you're finding results that don't matter? And then, when you're thinking about that, what is the biggest risk of it being too small of a sample size or too large of a sample size when you're interpreting longevity science?
That's a major problem. Okay.
I bet.
And no, it is. It's because too many people, it's like tossing a coin. Or
Yeah.
let's say a dice. How many times do you have to toss it to get a six? Well, a lot of
So many
scientists
times.
that don't have any background in statistical theory will say six times. Okay. But
Yeah.
to be 99% certain that you'll get at least one six, there's a formula that you could calculate that I'm really good at, but it typically when numbers get big, it always becomes 4.6 times that number. Okay. So if you figure out what's the probability that an event will happen, let's say
Yeah.
a particular drug will cure a certain disease, if you have some minimum number on what is like, how many people, so how many people do you expect to see cured at the worst case scenario? Okay. So you get that, let's say you expect to see 1 in 20. Okay. Well, you have to test 4.6 times that many people. Okay. And it turns out to always
Sure.
be that number unless it's really low. Like if it's like you expect 50% of the people to be treated, then you only have to multiply that by 3.3. These are numbers that I just have used
Yeah.
them so many times I remember these. But typically when you get into things that have a low chance of success, let's say lower than like 1 in 10, it's always 4.6. So multiply. So in order to do a study pretty well, if you think the worst case scenario is X, or let's say a parameter A, where you 1 in A number of people are going to be show a positive sign. If they don't, it's a waste of time to do the study, let's say. Then
Right.
you need to do 4.6 times A, and then have that number of people in your study. I just can't believe how many scientists I have worked for me and things like that will come and give a report at one of our scientific staff meetings and say, such a thing doesn't work. Look I tested it 10 times and got no hits. And everybody would just be all over the person because of the fact that they did about 5 times less than they should have done. That kind of thing.
Right.
But that's a really, really big problem. So many studies have been misled because of that. But again, it's hard to do the right number of people in a clinical study, so you have to do the animal studies. And a lot of times it's very okay to do a clinical study with just one person. Okay, if you have good markers, especially since the cost of doing the studies are so expensive a lot of times.
So expensive, yeah.
And then after you do the one person, then do another person, and then do another person, and just build as you go. Okay, but again, you're not going to be able to do double-blind placebo-controlled at that time, but you just have to have good markers to make certain. But that's a tough question. It's just a lot of studies are meaningless because they didn't test enough samples.
Yeah. Well, and I'm sure like other things play into a factor, like you get a sample size full of like 50 people and they're all white women age 32 that grew up in North Carolina, and they all have different genetic markers. Like I don't have a gray hair, but I have many peers that are my age that grew up in North Carolina and grew up in similar standing as me that have gray hair. And so our results are going to be different. And so I'm sure that plays into it too, where you're like, oh, well, this person is not going to have effective data because of this part of their genetic makeup.
People in the biological industry field do not get the proper training in statistical analysis and statistical theory. And that's why I'm so glad that I got a degree in experimental psychology, because everything you were just describing is exactly what experimental psychologists would take into account and try to factor out as much as possible, and sociological too. So even in psychological studies, you have to be sociological too, in terms of demographics and things like that. And that's something that's extremely important that 95% of all biological scientists don't understand. Okay, so that's one of the reasons why I decided in my quest to learn everything I could to understand why we age, how we age, what aging is, and how not to age, I chose psychology. Experimental psychology is one of the studies that I needed to really do well in.
Yeah. Well, Dr. Bill, I know that you have got to pop off and do another podcast because you're famous. So I just want to say thank you for having this little chit chat with me today. And I look forward to doing it again soon.
All right. Well, thank you. And see you next time.
See you next time.
Thanks for joining us on Up One. If you found today's conversation valuable, be sure to subscribe and share this episode with someone who's curious about the real science behind health.
Have a topic you want us to break down? Send us your questions. We're here to help you separate fact from fiction.
Until next time, stay curious, stay informed, and let's keep taking it up one.